There we go. We've got people joining, which is fantastic. And we're just going to allow a few more minutes for people to come in if they're just a little bit late. If you're joining, thank you very much for joining. Yeah, so we'll kick off in a short while. But okay, so we'll give it one more minute and then we'll kick off. And just a quick thank you to everyone who's joined already. Please bear with us for a second, and we'll get going. We're 4 minutes past, so I think we'll make a start. So good afternoon, and thank you very much for joining us on this what is a beautiful sunny day today for the 9th in the Meaning of Light series of talks. And of course, a huge welcome to Dr. Geoffrey Guy and Alistair Nunn of the Guy Foundation for joining us. So thank you. Just a little bit about the series. The Meaning of Light series is a series of talks exploring the topic of light and our connection to it. And today we're going to explore origins and why light matters to human health and aging here on Earth and in space. So we have the pleasure to have Geoffrey and Alistair with us, who've been working in the field of quantum biology and who published a paper titled The Health Hazards of Space Travel. And within the study, there was a significant section on the topic of light. And that got me thinking about what the differences are between the lit environment on the space station versus our internal artificial light environments back here on Earth, considering we spend roughly 90% of our time indoors. Geoffrey, in your recent book, which I have here, which you can't see, you can see now, the book Quantum Biology, you wrote, if there's a single entity that's played a bigger part in the quantum biology story than any other, it is surely the photon. And if there's one area of research into the medical applications of quantum effects that has grown most quickly in recent times, it is photobiomodulation, the therapeutic use of various wavelengths, intensities and oscillations of light. So I'm really excited to hear about light beyond vision and photosynthesis, and how maybe we should be considering the use of light as a mechanism to deliver health outcomes for humans rather than as a therapy after the event. As we've done on previous talks, we welcome questions as and when they come, so please don't hold back. Fire the questions into the Q&A area and we'll tackle them at the end of the presentation. Alistair will be presenting. And so with that said, let me hand over now to Dr. Geoffrey Guy and Alistair Nunn. Geoffrey, why don't you start us off by telling us a little bit about yourself and the background to the Guy Foundation, if you don't mind.
Well, thank you very much indeed, and it's a real honour to be able to speak to you and your colleagues today. I've noticed there's a couple on board that we know already. So I'm a physician, and I've been involved in medical research, mainly in the pharmaceutical industry, for over 43 years now. And during that time, I founded a number of pharmaceutical companies. 3 of them were publicly traded here in London and on Nasdaq. And throughout that period, I and Alistair, and I was a co-author with Alistair on a number of papers, over 80 papers, 3 books, and in fact, I'm the inventor on 134 patent families extending to over 800 patents. So I've been quite busy in research for that period of time, but I'm probably best known in the last few years for my work on cannabinoids, and that is, I was asked by Her Majesty's government to see if I could sort out the cannabis as a medicine issue back in the late 1990s. And I and my team, we developed extracts of cannabis to treat multiple sclerosis, and more latterly, we did a lot of work on something called CBD. I think a lot of people might have heard of that, in catastrophic childhood epilepsy. And both of the products involved there we had approved around the world, including the United States. So Alistair and I, we go back a long way, but for about the last 20 years, we've really been looking closely at intracellular bioenergetics. That's the way in which the cell produces, manages, and manipulates energy, not only for motive force and growth, but also for balancing other homeostatic mechanisms that go on in the cells and in the body. And when one looks at that, one is drawn entirely towards mitochondria. And now mitochondria, Alistair is going to take you a little bit more about that. But when I was at school, and many of your listeners were at school, you probably think of mitochondria as part of biology, but the more we look at it, the more we see it as a mathematics and physics construct. And we moved our thinking from biology through thermodynamics into quantum biology, and Alistair will explain that in his talk. So I used to describe myself as a full-time medical student with a giant-sized chemistry set. Now I have to describe myself as a full-time medical student with a giant-sized physics set instead. So we established, my wife and I established the Guy Foundation Family Trust in 2018 to begin to research and promote the research in the area of quantum biology. And for your listeners, quantum biology is basically the impact of quantum mechanics on biology. And quantum mechanics is the sort of physics you didn't learn about at school. It's the weird, very weird stuff of very small and very quick, minute subatomic particles, which of course can also be waves and particles at the same time, and events that occur in things like 10 femtoseconds, 10 times, that's 10 to the minus 15 seconds. So very small, very quick. And it used to be thought to be very cold and very dry. But of course, we now know that quantum effects do occur in warm, wet organisms like humans. So today, we're going to carry on from a theme I think that you and your colleagues have spoken about. I know Bob Fosbury has spoken to you about the emerging understanding of the health hazards of the lack of infrared light and the loss of the appropriate diurnal variation between blue and red throughout the day. And quietly over the last 30 years, we've basically started to starve humans of red light. We've replaced our incandescent bulbs with LEDs. We've stopped sitting in front of open fires or even open electric bar fires. And we now insulate our properties with glass that doesn't let the heat out, but actually won't let any infrared light in. And so this is something that there is a health hazard. And Alistair is going to explain to you the underlying rationale as to why this is actually rather more serious health hazard than might have been thought of heretofore. So without further ado, Alistair, if you would like to put your slides up, and do be gentle with the audience, Alistair. Put your slides up and give your presentation.
Okay. Right, there we go.
There we go. Perfect. Amazing.
Perfect. Yep. Sorry. It's just because I was trying to look at the camera and look at the screen at the same time, and it doesn't allow me to do that. So, okay, I'll try and look at both. Thank you very much for that great introduction, Geoffrey. Okay, off we go. As has been discussed, I'll try and keep it relatively light in terms of the deep science, but hopefully there's some concepts in here which everybody will kind of understand. As Geoffrey said, the background to this, and this is quite an interesting one. Geoffrey and I worked together, started working about 18, 19 years ago. And we were, when I was employed, to start thinking about how cannabinoids and phytocannabinoids, the plant compounds, actually work. And certainly then the idea that Geoffrey had was these bioenergetics. Anyway, given my background, I realized quite quickly that they modulate mitochondria. Now, the interesting thing about these compounds is they actually also fluoresce. And this led us down a very interesting path of looking at light and the effects on cell. And we soon discovered actually that the cell was full of these things called chromophores, kind of well known, but nobody really spoke to you about it. And during this time, we started reading around the subject. I started to read a lot more about mitochondria and discovered actually there was some very interesting research on, how they originated, which is very important, and how they evolved. And this led us down into some books by Professor Nick Lane. Now, some of you, if people are interested in mitochondria, read Nick Lane's books. They are absolutely brilliant. And in one of his books, he mentions this concept of quantum tunneling. And we went, oh, quantum mechanics, what's that got to do with biology? And that basically set us off down a whole new route, and leading up to working and looking at the effects of quantum mechanics and space health and so on. And here you'll see I've got a few graphs here. At the bottom here, I've put these two pictures at the bottom. This is, of course, Darwin and Stephen Hawking. And what is interesting, both of these were very interested in natural selection. And even Stephen Hawking was talking about natural selection right at the origins of the universe. And this is something I wasn't aware of until quite recently. So this is a very fundamental principle of life, but actually seems to have a lot of bearing in physics. And this led us on to this document here. My background, I'm a biochemist. I studied cell death. Why do cells die? And you'd be surprised that all cells die, commit suicide, including bacteria. Worked on cannabis, obviously. But my interest very much is in how we can use understanding of the origins of life to understand modern medicine and health. And in particular, why it is that we seem to have variations in how long people live and why the environment does this. And there are some really big questions that come out of all this. Now, there are some very big questions in biology. How did life start? What is aging and why do we die? These aren't fully understood. What is consciousness? This is the big one. What is awareness? What is cancer? We kind of look at it, but actually why is it happening? Are we alone in the universe? Of course, I think everybody kind of asks these questions. But the meaning is we still have a long way to go to understand biology. And this is interesting in terms of medicine, because medicine has largely been empirical. We don't often need to know how something works as long as it works. But that may be changing. We may need to have much more understanding of theory of aging and stuff to produce better medicines. And we kind of need a grand unified theory of biology. And physics could help us, but the really interesting thing here is that biology may be telling us about physics and maybe explain a bit more about quantum mechanics. There's a really interesting time to be doing this. And this, on the right hand side here, I've got this idea here. This is a galaxy. And the question is, of course, what about the physics of our universe led to people like this chap here called Einstein, who developed ideas which enable us to build something called the James Webb Telescope and to look back into the universe and ask the same question: how did we get here? These are very fundamental. And I've always liked this quote, and those of you may remember or have heard of Faithless, the musician. In one of his songs, he goes, "You don't need eyes to see, you need vision." And I think this is absolutely really quite a clever quote. So what I'm going to talk about today, try and keep it top level. I'm going to explain a little bit about why the origins of life may help us understand today's biology, a little bit about why quantum mechanics, thermodynamics, and this process called hormesis, which I'll come back to, and things like photons, electric fields, even flight envelopes can help us understand modern life. And why these things could explain why we may experience accelerated aging. And why in particular, and our little friend the mitochondria is a very good metabolic canary in the coal mine, why it tells us a great deal. I'm not going to talk about photosynthesis and vision because that's another 12-hour lecture, something which most people, but I'm going to raise this point here. This is the right amount, this is idea that wasn't really prevalent until quite recently. The right amount of sunlight means you live longer. And I'll come back to this diagram, but fundamentally, we're now realizing there's a lot more to sunlight than people realize. I'll explain it later. So why is light important in biology? Oh, this was supposed to move and it's not moving anyway. Just as vitamins ensure optimal function, and we all take our vitamins, we know they're important, it seems that various wavelengths of light can be called vitophonons. Now, I've just come up with this term, and I do apologise for it. It's probably not the best term, but a phonon is a definite discrete unit of quantum or vibrational energy. Why may be life dependent on these, or at least function better with them? And what's really interesting, and this is just these look like 3-methyl chicken wire, and this is very big chemistry, but these are molecules that can originate in the universe and are really key in how we work today. And all of these molecules absorb light. And these are vitamins, vitamin B12, E, etc. They all absorb light. So what's the connection between these? Okay, let's have a look at one of the things we're going to talk about a bit about space. And these are very recognizable people who went to the moon. And now there's a little problem which has been emerging in the space world. And this is that actually astronauts seem to be developing all sorts of symptoms. And several groups going back about 15, 20 years have raised the point: is going into space accelerating the aging process? This is not universally accepted, but there's a lot of science behind this. And one of the things which has been noticed in the last couple of years is the mitochondria are getting very unhappy. They're not functioning properly. Why is this? And it seems to have similarities to what happens when we have a very poor lifestyle, i.e. we eat too much and don't exercise. And indeed, multiple systems in the body are affected in space. Just about everything anybody's looked at shows there's a problem, ranging from your lungs to your heart to your brain to your gut, your bladder. Things start going wrong. Not enough to cause you to be seriously ill, but they are not functioning properly. And of course, the mitochondria causes unifying all of these. And those of you who don't know about mitochondria, I'm sure some of you probably do, but these are little things inside your cells. Now, the backstory to these is really important. Evolution and theories suggest now that the modern cell, we call this the eukaryotic cell, and what that means is a cell with a nucleus, evolved somewhere maybe 1 to 2 billion years ago, maybe further back in time, when two kingdoms of prokaryotes got together. One was a bacterium, if you've heard of those. The other is an archaean. You may not have heard of these, but these are just as important. And they had a love story. They got together and they created the modern cell, which basically means that the ancestor of your mitochondria is a bacterium. And there's been lots of evolution since then, but it means they're central to a lot of things, everything in the cell fundamentally. You will have seen them in textbooks saying, oh, we eat food, we extract electrons from food, those go down to water, they generate ATP. And this what's clear, very important here, they produce a proton gradient. But they control just about everything else in the cell too. Inflammation, adaptation to stress. They control whether the cells live or die. They control how fast you grow. They generate through this very ancient sort of chemistry thing here. It's what they call the TCA cycle, all sorts of molecules which can help you develop things like, build fat or build more muscle. But they also show something which is, again, fundamental. They generate heat. And this is through a process which we call uncoupling, which is basically dissipation. They dissipate energy. And people kind of think, well, why do they do this? Well, it's actually fundamental to what life is. And I'll come back to that. And one of the key takeaways from this is basically, as was said by this chap, Albert Sense Gordon, that life is nothing but an electron looking for a place to rest. Now, just think about that as we move forward. So here's the idea. Mitochondria are very good canaries. And if you don't have very healthy mitochondria, you seem to demonstrate this thing called reduced metabolic flexibility. It's basically, I think, spare capacity to deal with stress. Without this, you get ill much faster. And we see this with virtually every disease. There's a mitochondrial dysfunction component, and it's associated with accelerated aging. And it's actually, and it's impacted by all sorts of things. What are the commonality between COVID-19, this chap here with a fat tummy, and an astronaut? Well, all of these components could actually reduce your metabolic reserve, and this could be really important. Now, this comes from my background and the team. I work up in Westminster University some of the time with the team there that look at optimal health. We're trying to define what health actually is. And the group I'm working with has studied fat distribution for years and years and years, and they've been key in the biobank studies of visceral adipose tissue, for instance. But what we know is if you have less muscle and more adipose tissue, you tend to get in an imbalance. And muscle can be seen as being generally anti-inflammatory, especially if you use it a lot. Whereas adipose tissue, if you have too much, it starts to become inflammatory. So you get an imbalance. And what we know, this is associated with reduced mitochondrial or mitochondrial dysfunction. And we got this, what we call reduced metabolic flexibility, i.e. we can't deal with all the stress. And what you see in many modern populations now is an expression of this thing called the metabolic syndrome, which is increased visceral adipose tissue, insulin resistance, which can lead to diabetes, altered good to bad cholesterol ratios and high blood pressure, and inflammatory. You tend to become very inflammatory. And what it seems to do is accelerate the aging process so that a lot of diseases appear at a younger age, and it reduces your resistance to viruses. Sorry, this looks like the eye of Sauron. It's a terrible slide, but it makes the point. Even when we look at viruses, they modulate mitochondrial function. And this is why COVID-19 and mitochondria go together. It's worth understanding them. But what we do know is if you lead a very healthy lifestyle, and I mean by this, but a lot of exercise and the right diet, you get more muscle and you become more resistant to things like viruses. And it's not universal, but it seems to be generally the case. And you can resist all sorts of things, like you can deal with, for instance, more temperature extremes. And so it means you seem to be more adaptable. And this is associated with metabolic flexibility. So another way of looking at this, this is a slide we produced ages ago, but the green line, the solid line, represents what we think is the optimal life expectancy of the average person if they lived a very healthy lifestyle. This is taken from a lot of data, but critically, what you also see is this dotted line is the healthy life expectancy. And what seems to happen is if you live in a really good life and everything goes in your direction, you live healthily for many years, and then everything goes wrong at the end. And you get what we call morbidity compression. What we actually see, and this is the actuarial data of the world, is that the average life expectancy is between 80 to 85, women living slightly longer than men. But what we see is an expansion of the time and the percentage of your life that you're not as well. If you live a really bad lifestyle, this gets even worse. And there's evidence that causes your life expectancy drops even further, but you live a much greater proportion in ill health. And I was talking about this with Ulysse earlier on. This means the cost to society is huge. So living a bad lifestyle has a big cost impact. And in fact, you go too far, you can live a very short life. But it gets worse. We now know that there's this process called epigenetics, and that is that your parents can transmit some of what's happened to them by modifying the way your DNA is read as a child, and including our grandchildren. So potentially a bad lifestyle can predispose children and grandchildren to having further problems. And this is quite scary because it means that life is a vicious loop. And kind of what the message here is that environment actually has a very large component of determining how well you live and how healthy you are. And of course, the connection we're interested in, of course, is where does an astronaut sit in all of this? But in terms of data, there's a very interesting paper that came out a couple of years ago showing that they looked at they looked at the American population and said, well, how many people are actually kind of defined as healthy? And it may be less than 7%, which is quite scary. And just another way of looking at this, way we look at this, if you live an optimal lifestyle, you're born, you live fairly healthy all your life, go to hospital, don't live very long at the end and then die. What we actually see is this, and the poor lifestyle leads to this. And so the question is, where do astronauts sit in this picture? Okay, now, some of you may be pilots, some of you probably may not be, but certainly if you're engineers, you'll understand the idea of a flight envelope. And this is taken from a little light aircraft or something. But most people will know if you try and fly too slowly, it stalls. If you go too fast, the wings come off. And so there's an operating region. And life seems to have exactly the same kind of thing. We have operating limits. I mean, we can't live underwater or we can't fly. But fish can't live on land, you know, and birds can't, well, some birds can swim underwater. Point is that there is a range for everything, every life form. And what's interesting is that life can adapt. It can expand its this envelope a little bit. So we can actually modify this a little bit. This is what we call the metabolic flight envelope. And you'll notice I've taken out the F there. And the question is, you know, why is mankind suddenly seeming to have all sorts of problems? And there are more problems than people realize. And this kind of goes back, and I won't go into detail, but it goes back into what we think is going on, is that intelligence, and we like to think of ourselves as being intelligent, but we spent billions of years under a great deal of stress, evolving and evolving and evolving. But once we started to develop technology, we could take away all of those stresses which made our life difficult, like having to move, having no food, you know, having no shelter or anything. But what this seems to do is take away the driver for our adaptability. And it seems that this is associated with the decrease in life expectancy. And we've used this idea. We published this ages ago as a parable paper to say, well, you know, when we use this to explain one of the big questions about, are we alone in the universe? And what we began to realize, of course, is that if all advanced civilizations, just for a second, say they are out there in the galaxy, they may all end up spending so much on their healthcare that they can't afford space travel. Now, I know it sounds a bit silly, but actually, this is one potential thing. If all life tries to make its environment too comfortable, it actually shoots itself in the foot in the end. So this is the idea. And I would say, oh, was there any data for this? Well, yes, there is. This is a paper that came out about 15 years ago showing that obesity was costing something over $2 trillion a year, only matched by war and smoking. And these figures have been revised upwards as we go through time. And a couple of years ago, it was estimated that space research was costing globally is about 117 billion, but obesity could have been 2 to 4 trillion. And so this raises quite an interesting point: is that, you know, in the USA, space research was about 0.2 to 0.3 of GDP, but obesity was costing 2 to 3%. And this is causing problems because NASA doesn't have a huge budget. And within that budget is actually quite a small component, which is actually looking at health, really looking at health. And this is the BPS, the biology and physics section. And indeed, there's been publications on this saying we need to spend a lot more on trying to understand astronaut health. And this is this document called Thriving in Space. Just another way of looking at it. Yeah, I did this slide about 10 or 12 years ago. Makes the point that we spend a fraction of what we're spending on obesity. And I think it's quite scary, really. But anyway. So the thought for the day from this section is maybe that the ability of humans to expand beyond our own planet may well depend on our understanding of aging and health. And we haven't really cracked it on Earth yet. I think we know some of what we need to do, but we're not really doing it yet. Okay, let's move into some physics and the origins of life here. I'll come back to this little picture here in a minute. Key point, we are electrical. Most of us, you probably know, we generate between 80 to 100 watts of energy a day, but we contain something like 10 quadrillion mitochondria. They're all flowing millions of electrons a second. So we're fundamentally flowing electrons all day. And one of the things that these electrons do is generate what we call a proton gradient in our mitochondria. And these protons, it's a hydrogen gradient. And when you look at the actual charge, it's approaching 10 million volts per meter. Inside all of us, not only do we have a huge electric charge inside our cells, but actually this is connected to little turbines that are spinning around 2 or 3,000 RPM called ATPs. So there's a lot more going on in there than many people realize. Now, in terms of origins of life theories, I'm sure people are probably aware, there's quite a few of them. There's the RNA world idea. This is that little nucleotides which evolve very quickly could generate modern life. There's the fatty acid world, alkaline thermal vents, which is something I'll talk a bit more about in a minute, which we call metabolism first. So rather than DNA first, we have metabolism first. There's the purple earth. I know you'll be fascinated by this one. And there's this idea that the precursor rhodopsin, which is a key element in your eye being able to detect vision, actually in ancient bacteria, its precursor could take on light and generate a proton gradient. This is what they call an isoprenoid. So it's a molecule which could have originated off planet. So it's absolutely fascinating. We have UV light theories. There's something called zinc sulfide, but people thought could be important in thermal vents, or complex chemistry in oceans. And there's protein first polymers. And then, of course, there's Darwin's warm little ponds and basic thermal vents. And of course, the big theory that all the science fiction fans love is panspermia. This is the idea that life came from somewhere else. And of course, Star Trek does this, but of course, it only puts the problem somewhere else. Now, just moving a bit more into some of the photon stuff. What did life need to get going? Well, what we now know, and sort of Bob will talk about, he could mention some of this, but we now know a lot of organic, what we would call organic molecules, carbon-based molecules, can be produced in interstellar clouds, in planetary disks, right through the galaxy, well before there's any life. And a lot of these things have these kind of structures, and I won't go into the actual structures, but some of them look like vitamins, i.e. there is already existence, long before life got going, a smorgasbord of chemicals. And a lot of these are generated through light and heat, but there's some very interesting chemistry. Now, quite recently, there's also been this idea. Now, there's a molecule in the cell called ATP, which is key in moving energy around the system. And somebody's predicted it actually could have been generated in volcanoes. And this is actually, I mean, if it's real, it's quite amazing, just through basic chemistry. So it's suggesting something else. What we now know, and what we think we know, is that ATP isn't just an energy molecule, but it affects water. And I won't go into that now. So we have to start rethinking how we view even basic molecules. So, of course, there's still a big question is, okay, we have all this chemistry, but how did life start? Well, it needs to be organized. And this is where thermodynamics, and what we call adaptive thermodynamics, come in. The basic principle here is we know from maths that if you have a mix of molecules in an energy gradient, they will self-organize into structures which accelerate the dissipation of that energy gradient. And the classic example of this is, say, a hurricane. A hurricane has structure, but it comes about as a means to dissipate energy. And this seems to be a fundamental principle of the universe. And self-organization of these structures could be something that began life. And what it seems is that nature hates a potential. And this was kind of taken from Schrodinger, but life is basically a piece of negative entropy. It's a structure that accelerates the heat death of the universe. Now, that some will completely disagree with that, but it's a really interesting idea. We burn through a lot of energy. So what else are we missing? Are we missing anything? Well, we have all the chemical starters, electrons looking for a home. We have good theories on how they might organize. We even have theories on how the information can be incorporated into this. So there's some good maths behind this. But we also have natural selection and survival of the fittest, which comes from the ideas of Darwin. And we also have lots of energy. And the question that comes out of this is, well, that's all very well, but we all know that we have we know we exist, we're self-aware. Did that require a starter at the beginning? For instance, did we need a crystalline cavity or a membrane vesicle, like a little fatty acid bubble? Do we need a clay surface? All these things. Do we need fields? No. Did electric fields come into this? Something to do with water order? These are still things we're not fully understanding. But when we start to look into the sort of cupboard of mysteries, we discover actually when we start to look at the origins of life theories, there are some areas which have not really been explored. Electricity in fields. This is clearly something which is really important. Photons, not really, apart from driving basic chemistry, but do they have another gravity is another one. magnetism. And I'm not going to go into gravity or magnetism too much, or electricity field, because they get really complicated. So let's put some historical context behind this. You'll see why the onions and the Tottenham light bulb are important. During the 18th and 19th century, a lot of science, people started to really investigate. And one of the things that people began to realize was this thing called electricity. And Galvini and his frog's legs was one of the key aspects, but Voltaire came along as well and looked at this. But this led to ideas in the late 19th and 20th century, and the beginning of the 20th century, that this could be this thing called the morphogenetic field, because nobody could explain why your finger was a finger, a frog was a frog, a giraffe was a giraffe, or a human was a human, and how we generated and how we grew. What was the, how did the leg know to be a leg? And so people were starting to think, well, there must be some kind of ethereal field. And one of the key characters in this time, around the early 20th century, started to think about this. And this is a chap called Alexander Gurwitch. And quite why he did this, I'm not entirely sure, but he decided to look at onions. And what he did was cut a root of an onion. And this was in the early, the early 20th century. And he thought, well, I'm going to look for something which isn't chemical. And so he had another onion and examined its growth. And what he found was that if you cut the root of an onion, it transmitted a ray through this, like a quartz barrier, and caused these roots to start growing. He called this mitogenetic radiation. And he showed that this was UV light. Now, that's pretty profound if you start thinking about it. And this has led on to some really interesting science, and there's big legacies. And there've been many scientists, both past and present, looking at this. And why is it biology seems to be producing what we call biophotons, or what they call ultraweak photons, and non-chemical communication. These are just some of the key characters who've been involved in this. But at the same time, people are also thinking, well, if there's electricity involved too, is that important? And this is and so there's this thing called the bioelectric field, and this goes into electrodynamics. But the point about this is we can start to look at this and go, well, actually, there's this thing called bioelectromagnetics and photobiology, and they're linked through quantum mechanics into explaining potentially what life is and how it works. And this is now becoming quite accepted in scientific fields now. We're still trying to develop it, but it's becoming. There's a really nice book by Fels, Siffer and Schockelman, which you ought to read, which basically just summarizes all of this. Now, we throw our full penneth worth into this and then go, well, okay, so say we've got a morphogenetic field, and this comes from the work of Mike Levine, who shows that actually you can control regeneration in a variety of organisms, not by changing the DNA, but essentially changing their bioelectric field. There's things called ion channels you can play with, which completely takes DNA out of the picture. I mean, it's absolutely fascinating, and this has been very heavily looked into now, and it could be the way we're going to understand how to generate new organs. I mean, this is the kind of significance of this. But we looked at this and, well, how could this happen? Well, the interesting thing is, I mentioned these things called alkaline thermal vents. And this is one theory about how life evolved. And these are vents found in the deep sea, and maybe there've been a lot of them in the Hadean Earth. And what seems to happen is that through a process of geochemistry, you get a lot of hydrogen upwelling, and it meets alkaline acidic seawater. And this generates some very interesting chemistry. But it also must be generating fields, because you've got a flow of charge. And we suggested that this charge could have been something that led to organization. And when you start to put in various other theories like water catalysis, you can start to see, if you start mixing up all of these chemicals, you can start to see where life could have started. And critically, this could have been independent on fields. Now, what we also now know is that in modern cells, we've got this thing called a cytoskeleton, and there's a lot of literature on this, but the cytoskeleton are microtubules. It's like the inbuilt skeleton inside your cell. It's very sensitive to light. And we think there's an interaction going on here, but the outcome of it is you generate this like a field, a morphogenetic field. So this could be how it started, but clearly it gives a definition of self and non-self. Now, are there any smoking guns? Well, of course, we can argue that bioelectricity is a smoking gun. But you remember I mentioned this thing called uncoupling. Well, as it turns out, all cells demonstrate what appears to be futile cycling. They cycle through loads and loads of ions. They're pumping ions and then letting them back through. It's like trying to paddle uphill and like you're having a dam, but keep letting the water flow back through it. But this is central, because what this is dissipation. These structures constantly dissipate ions. They're generating energy, heating up all the time. But all organisms, including bacteria, do this. They release 20 to 30% of their energy in this futile cycling. But of course, when you think about the theories of organization, this is absolutely essential. So the smoking gun could be this. And what we know is that cells have very careful systems to control, like a floodgate. They can open these floodgates to control the system because it can get overheated. And so this seems this is part of a system to maintain a structure of a cell. And it basically suggests that, I won't go into that, it's complicated. But of course, you're all thinking, okay, that's all very well. What about photons? Well, we can start to look at the same process with photons in a way. And again, we go back to our universal soup idea. We end up that universe generated all these various interesting chemicals. But of course, when you introduce light to this, you start to induce a energy gradient, which causes a chemical cascade, which absorbs and dissipates the energy. And this can actually also self-organize. And of course, what we know is that organisms from very early on in life develop these molecules which can act as sunscreens, trying to dissipate the excessive energy. And of course, modern cells, and particular plants, produce huge numbers of these things, which started out as sunscreens, but of course they have all sorts of other functions. And the interesting thing is, when you start to look at this, you can start to go, well, why is it these compounds exist? And this goes back to the work I did with Geoffrey. But we've been trying to understand, well, why would a plant compound, now you all take aspirin, you all take resveratrol probably, and turmeric and all these things, and you're asking the question, but why is it they work in a human as to help you feel better? Well, this could come back to light in the end, because these molecules, a lot of them can sit in a membrane, they dissolve in a membrane, and certainly in biology, your membranes have to be controlled very tightly in terms of their fluidity. If they're too stiff, they don't function. If they're too leaky, they don't function. So biology spends a lot of time trying to keep your membranes in the right spot. And what we know about these compounds, which their structures are ancient, is they can dissolve in a membrane, but they can also alter the fluidity of this membrane. And they're also antioxidants. And one thing that really messes up your membranes is getting oxidized. That makes them very stiff. And temperature obviously goes the other way. So there's an interesting corollary here, but maybe these are compounds also interacted with light to maintain your membrane fluidity and thus dissipation. We don't know, this is a guess, but it's worth thinking about. Now, the other thing here, and this kind of falls out of what I said earlier, is most of the key molecules in how you generate energy all absorb light, and many of them are fluorescent. Basically, these are chromophores, and your cells and your mitochondria are stuffed full of these things. And these are just, some of you might be familiar with these spectrums. This is the absorbance spectrum for the key molecules in your cell. There's key ones that are called the flavins. NADH is a really key one, but there's amino acids which and these compounds fluoresce and they re-emit light. And the point I wanted to make here is, look, they all overlap. Is this actually something which is just serendipity, or is this a dissipative structure of light coming in and slowly being dissipated down a whole chain of electrons where you're generating electrons? It's a really interesting point. And I just raised this thing. Most of you will have heard of salicylic acid or aspirin. But salicylic acid is a plant hormone, and plants use it all the time. And it has some very interesting properties. One, it fluoresces, so it dissipates light energy. But it also uncouples. If you put this into cells, it uncouples membranes. So why is it doing that? And most of you will know it, other than taking aspirin, it's used in sunscreens. If you link it to a large polymer, it becomes a good sunscreen. And you've all wiped it on your face at some time. So I don't know if you've seen the connection with aspirin, and this is how it absorbs light. So this is this constant connection here with absorption. This is just another look at some of the more key molecules. These are all 3-methyl chicken wire, I know, but the point about all of these are all absorbed light, are essential to how we function. And this includes DNA. Now, a lot of you think DNA, it's all about just genes, but actually DNA has another really interesting function. It's a very good sunscreen. And guess what? Bacteria use it as a sunscreen. And we now know, and this goes back to very old times, if you get these biofilms of bacteria, and some get damaged, they commit suicide and release their DNA to protect the other bacteria from UV light. So you kind of got to think differently about what you're looking at here. And these are just some of these other key molecules. And these are all molecules you'll find being sold on the shelves of various nutraceutical departments, resveratrol, genicin, green stuff in green tea, quercetin, berberine, curcumin, cannabidiol, etc. All these molecules can absorb and emit light, but they can also modulate mitochondrial function. So that's all very well. Now, I mentioned this thing called biophotons. Why is it cells emit photons? Well, we now know that cells during metabolism emit very low levels of photons. So they glow very slightly. We can't see it because it's way below what we can see. but instruments can detect it. And this is what Gerwich discovered. And we now think that they're produced doing day-to-day metabolism. And the key thing here is that it's related to oxidative stress. And I won't go into all the chemistry here, but if you stress cells, they tend to produce more of these photons. And this suggests something really, really interesting, because it may indicate that they're actually, if you've got a cell producing its own photons, and this is not bioluminescence, which you see in, say, fireflies, it's actually internal to the cells. Is this part of a system to how the cell functions? We don't know yet, but it seems like it could be. And this is what we now know, is that we're looking at this, and we know that the wavelengths of these photons goes all the way from the UV up to the infrared. They're very low level, but they're there. And this suggests, actually, we start to think about this concept, what we call photonic homeostasis. Is there a feedback between the chromophores in your electron transport chain, for instance, and your metabolic photon? Don't know, but they could be. And this is all this could suggest this is part of dissipation. And Darwin would have understood this. Like, nature never wastes anything. Why would it have not used biophotons or photonic light and electromagnetic fields as part of a homeostatic system to maintain that system in its best state? It's been doing it for a long time. So maybe we're missing half the story here. We're missing the role of charged particles, the role of photons, and this idea of morphogenetic fields. And we know that if you lose your membrane potential and stop flowing electrons, you die. So this is all part of the same process. Anyway, let's go back to space a bit. Recap. Mitochondria in space, they're not happy. What's going on here? There's lots more data coming out on this now. This is really interesting. And even after 2 or 3 days, we're seeing that mitochondrial function seems to go slightly wrong. So I think most people will probably accept that, this has been looked at, that it's a lack of gravity, maybe the increased radiation, and altered circadian rhythms. But if you think quantum, you start to think of other things. Well, what about direct metabolic consequences of a lack of a magnetic field, or certain wavelengths of light, which is what Bob Fosby has been talking about? And we worse when we go further from Earth. This is obviously Sputnik to the shuttle to Artemis program, which is going on now, and of course, whatever happens after that. Now, I'll just whiz through these. And some of these, I think the architects here will probably get this. One of the key things about lack of gravity is that life has evolved under 1G. You take away the gravity and the system doesn't fall to pieces, but it doesn't know quite what to do, because we rely on the compression of gravity. And we know this is linked to mitochondrial function. And all mitochondria in your body talk to each other. It's just not in cells. Cells swap them. And if you exercise, you improve your mitochondria right across the body. There's also potentially, and this is, again, we're not entirely sure what's going on here. We know that circadian rhythms are linked to light, but there's some evidence that organisms could detect fluctuations, either magnetic field or even the gravitational change with the lunar cycle. It's some very interesting physics in that. And the key takeaway message here, of course, is that mitochondria are integrated with your cellular cytoskeleton. Alter the pressure, alter the gravity, you're going to alter your mitochondrial function. And the key thing here is that we've evolved in 1G. Most organisms do not experience reduced gravity, because it's generally lethal. If you fall off a cliff, you're not really going to evolve very quickly. We've looked at this, or we looked at, I say, scientists have looked at the role of centrifuges in space, very clearly beneficial. The next thing is magnetic fields. Again, could be a link here. We know that if you live on Earth, that you're protected by the magnetosphere. But space is a really nasty place. And you've got a lot of solar particles coming from the sun, very energetic, protons, for instance. But you've also got these things called galactic cosmic rays. And these are particles, and they're actually things like iron nuclei, carbon nuclei, produced in stellar events. These are these are ions and large atoms traveling at near the speed of light. If they hit you, they do a lot of damage, and especially if you get a solar storm. And we all had some good solar storms, because I think most of you have probably seen the aurora over the last couple of years. We're in a high sunspot activity. The key thing here is that radiation affects the whole cell. You'll see people talking about radiation is damaging my DNA. That's just one aspect. It damages everything. Damages your mitochondria, it damages your mitochondrial, have got their own DNA, but it damages other big complexes in your cell. And when we start to look at radiation, this blue line here is a general average that most of us are exposed to, which is it's measured in this thing called millisieverts. It's 1 to 3 millisieverts. An abdominal CT scan, you get 8 to 9 millisieverts. The recommended yearly dose is about 11. ISS, you're up at 80 to 90. Going to Mars over a 180-day trip, you're looking at only 200. So going to Mars, you're going to be exposed to a lot of these things. And if you get an SPE, what they call a solar particle event, or a coronal mass ejection, it becomes very dangerous. Now, there's another aspect here, again, which may be related to photons and electrons, is, of course, when you go into space and beyond the magnetic field, you're exposed to much lower levels of magnetic fields. And we now know that biology is very sensitive to this. Birds can navigate using this, but we also know, and this is predictable from quantum mechanics, because of something called quantum electron spin, this is going to affect biology, and this seems to be the case. This is an old idea. The Chinese mentioned this a long time ago, saying, yeah, it's all very well that the lack of a magnetic field may increase radiation, but what's it going to do to biology? Now, the Earth has a field strength of about 50 microtesla, but when you go beyond the moon, it drops down to below 1 microtesla, down to 10 nanotesla. And you think, well, so what? But people have looked at this now, and you can put organisms and cells inside tubes which contain no magnetic field, because you can shield it, and indeed they start to go wrong. They show also, in particular, you generate reactive oxygen species, free radicals if you like, from the mitochondria, and there's lots of data on this. Proof of the pudding: if we alter it, we can modulate organism function, and this has been shown by several groups. You can say, for instance, change how stem cells grow. You can increase ROS, suppress growth, increase growth, and in fact even inhibit or regenerate growth. And people are now using it, for instance, to inhibit cancer growth. And of course, we come back to light again. And so the space thing, and this is a really interesting one. As we said, we're releasing this subject. Now, I think we're all pretty familiar with the red light idea now, and that this red light is clearly really important. And there's all sorts of studies now showing, ranging from how it can improve stroke victims, it can improve, say, glucose levels in the blood, and we can obviously accelerate healing. But what we also know, and this is the key here, it's not just red light, but all frequencies of light have an effect, because there are these many chromophores in the cell. So we've got to think slightly differently. We've looked at some of this now, and we've shown, for instance, that you can, this is a repeating experiment, if you put mitochondria into a little cuvette all by themselves and seal them, but measure how fast they use up oxygen, because that's what they do, you can show that if you inhibit one set of mitochondria, they send a stress signal, which is picked up by the neighboring cuvette. This doesn't seem to be chemical, it's light. If you put a shield here, that set doesn't do anything. So this suggests that these stress mitochondria are sending signal to others. We showed, so you can start to look at these ultra-weak photon emission, and we can detect it. It's actually very difficult to do because there's a lot of strenuous light. The detectors we now using, we're doing this up at Harwell in Oxford, near Oxford, can have a detector that you can put inside a box in a room, a dark room, and if someone turns a light on two doors away, it still detects it. Photons, they get everywhere. We've also shown that, of course, one of the things we can do is suppress senescence in cancer, induce senescence in cancer cells. So there's something very interesting going on here. And of course, we'll all be familiar with Bob's work. This makes the point, and this is the idea that infrared starvation. And certainly, if you look at the green thing here, this is what we see with LEDs, but what we actually receive on Earth has a lot more infrared. And I think this is key, which I'm sure everybody's familiar with. But I think it goes further than this. Now, one of the factors that everybody's talked about for a long time is this compound called melatonin, and this is quite a good example of how biology sometimes has to do a double take. For a long time, it's thought, well, melatonin, it's produced in your brain when you shine a blue light, and and if you look at a blue light at night, it suppresses your sleep. But that's only a tiny fraction of what it does. Melatonin is produced by all your cells, in particular by mitochondria, and maybe it's stimulated by infrared light. Of course, this makes the point to say, well, okay, well, what is melatonin? Well, the sense is that it is very, very ancient and probably evolved when photosynthesis, or even before, as a molecule protected against light damage, because it's a very powerful antioxidant. So we start to have to think a little bit differently about this. So infrared light, it could be doing a lot more than just what we think it's doing. But the other story here, and this is the one I mentioned right at the beginning about this slide here, is the UV mortality and vitamin D story. Now, I think everybody's aware that vitamin D is good for you, and you need sunshine, but there's also this thing, oh yeah, but too much sunshine induces skin cancer. Well, actually, it's not quite as simple as that. This is just a vitamin D, this is how vitamin D is made, but there was a study done in Sweden a long time ago, well, not that long ago, where they looked at the sun exposure and mortality, and what they actually showed was very surprising. People who were exposed to more sunshine Yes, they developed more skin cancer, but they lived longer. And I think this is absolutely fundamental because it seems they developed much less cardiovascular disease. And it seemed to be tightly correlated with the light they exhibited. Yes, they got more cancer, but they didn't die of it. They were dying of something else. And it suggests that not only is the dose key here, so very high levels of UV we know are damaging, but there's an optimal level of UV. And this is, if you get the right dose, the Goldilocks zone, as it were, it's good. But there's also evidence now that there are other things going on in your skin. For instance, there's some evidence that, for instance, UV produces nitrates in your skin, and this lowers your blood pressure. Take-home message is more going on than we thought. Other things from space station, circadian rhythms, I think you're all very familiar with this. Life is all about vibrational resonance. We all resonate with something. These are just Huygens clocks. But the point about this is that we now know that mitochondrial function and aging are tightly linked into those pathways which control circadian rhythms. If you alter those, you alter the aging process, and you alter mitochondria. And there's this lovely little paper that suggests that mitochondria also need to sleep. They need to, they need their 8 hours too. And so this could actually be very important. Of course, on ISS, the International Space Station, you're doing 16 days per day. Disruption to your microbiota. We all know we've got microbiota in our guts. Well, guess what? They talk to your mitochondria chemically. So because this is an integrating holistic process, there are far more of these than there are your own cells. And we now know in astronauts that their microbiota change, and not for the good in some cases. So clearly something's happening. And the other one, which again may come into buildings, I don't know how much it comes into buildings, but they're now finding evidence that in the space station, although they scrub the carbon dioxide out, they don't scrub as much as they should do. And there's now evidence that even small increases in carbon dioxide, even if they're not completely toxic, start to increase oxidative stress and maybe generating more mutations. And they did this, they put mice on the space station and discovered it wasn't to do with the lack of gravity so much, but the increase in carbon dioxide. And one of the things that we're now realizing, the aging process is related to mutation in your somatic cells. And these are non-germ cells. So not your gonad cells, but the everyday cells in your body. And they start to mutate. And as they mutate, things start going wrong. And so maybe this is accelerating the process. So what does this mean? Well, it kind of suggests that certainly in space, we're outside our evolved what we call canalized envelope. We evolved in a set of circumstances to survive in a certain set of conditions. As soon as we get out into space, we put us and any organisms completely outside our Goldilocks zone. There's a just right zone where we're all very happy, but you change the gravity, you change everything else, and it's not the right place. Now, the key here is okay, that's all very well, but what does this suggest about the aging process? Well, if we go back to thermodynamics, we can make a prediction that if you put an organism outside of its what we call natural selective zone or its Goldilocks zone, this is this idea of hormesis, where small amounts of stress induce adaptation, too much kills you. I mean, this has been around for a long time. It suggests there's a thermodynamic reason, and one of the reasons is potentially related to natural selection. And this is that thermodynamics suggests that systems only survive if they're ones that work best. And if you go outside of that level, they tend not to work. So natural selection is going on all the time in you, from your molecular all the way up to you, and in fact, almost at the level of species. And we know this. And if we look at young organisms, for instance, when you're young, you can resist a lot of stress. But as you get older, that process, you lose that process, and eventually you can't adapt at all. And this seems to reflect evolution, that when we started to evolve, and this is a long time ago, adaptation happened through genetics and simplicity. Think of your bacteria sort of multiplying really quickly. But with very complex organisms, we work very well because all our cells cooperate and become very complex. The downside is that if we go outside of our envelope, we don't function very well. And this results in electrons not going where they're supposed to. So we generate reactive oxygen species. So the idea is death, survive, adapt, survive, evolve. And this kind of scale invariance to do with quantum mechanics. And kind of what it means is we have to think about inflammation and death, at a global scale. If life emerged from the planet, that means everything is disposable as long as life survives. And smaller and short-lived organisms will survive. Complex organisms take note. And this is perhaps the best way of showing these are the diagrams from Darwin's book. But we know through evolution, things got complex. But when we have an extinction level event like that, everything has to revert back down the tree again. And then it re-evolves again. Think Gaia. So if we think about the metabolic flight envelope, Well, what happens to space? We're outside of it a lot of the time. Not completely enough to kill us, but it makes us not function properly. And it suggests that we're trying to evolve, but we can't because we're a single organism and we have to adapt. And this adaptive thing may be much bigger than we realise, and it's to do with oxidative stress. Again, just another way of showing the same thing. We evolved here. This is what happens in space. Everything's in the wrong place. So what are the conclusions from this? Quite a few, actually. The light, fantastic. Nod to Terry Pratchett, was his birthday, would have been his birthday quite recently. Humans are partly dependent on and are modified by external light, with a Goldilocks zone depending on time of day, wavelength, and intensity, where the hormetic response is key. This is adaptation to stress. Just the right amount means you're stronger, too much goes the other way. It's quite possible that we don't know, and this is pure speculation, but there's evidence it could be the case. We're relying on an internal photonic homeostatic system, maintaining your metabolism and everything, which is shielded to some extent from the external environment, but is also robust to some degree. It could reflect how life started. But the question is, how dependent are we? Now, thinking about space, and I've tried to do this, and this is I guess, this is the hierarchy of factors. If we think about where we evolved, microgravity puts us well outside where we should be. And the key thing here is that gravity is a key stimulus for mitochondrial function and reserve. Think exercise. If you don't exercise, your mitochondrial reserve drops. In space, that problem is 100-fold worse. Radiation, it decimates your mitochondrial reserve because you start damaging everything yourself. And if your mitochondria don't work, you can't repair the damage. Hypoxia, this is the carbon dioxide story. The hypermagnetic field, probably in the wrong place. Red light, I think this is really important. It's in the wrong place. The ISS, from what I understand, has now gone mostly to white LEDs. You're in the wrong place. Circadian rhythm's clearly a problem. How do we mitigate this in space? Well, I think it's fairly straightforward. We need to have a centrifuge, and we need to exercise. We need the right magnetic field. We need the right light spectrum. We need perhaps the right kind of diet, and definitely shielding and the right oxygen to carbon dioxide balance. You will see in many textbooks, they say, well, the obvious way that humans can expand into space is gene therapy. Well, spoiler alert, we're a long way from having decent gene therapy. That's not going to work in the next 20, 30 years. What this means for space travel? Well, it's quite serious. We know that certain people are trying to get their spaceships off to Mars fairly soon, but it's quite possible that actually astronauts, unless we dramatically change the spacecraft, are going to be quite ill when they get there. And what might actually happen is that a microbiota might evolve, but they won't. They'll just be very ill. These must jump in the gun. Of course, what does this mean for people on Earth, which is what we're more interested in? Well, in terms of light, we need to ensure our environment mimics the photonic flight envelope. And we evolved over millions of years. We probably ended up in caves with fires. Then we, of course, there were glaciations and things which probably stimulated adaptation. Then we had our huts. Then we, in the Middle Ages, developed our houses. But even in all these modern brick houses, we still probably had candles and fires. But of course, in modern day, we go from blue light. So this suggests that we've shifted from our ancestry through there to blue light. So what are the stresses on Earth? Well, again, I think we have to think of this differently. I think one of the key losses of stimulus is physical movement. We need to move. But I think the next one could well be light. If we totally change the light, suddenly we're building on a bad system. Circadian rhythms, clearly important. Other things like magnetic fields, hypoxia, and radiation are probably less of a problem. And so we kind of need to think about this idea of quantum hormesis. Well, I call it quantum, but it's about this adaptation. Complex life has evolved sunscreens to block most of the shorter wavelengths, and we probably all know that short wavelength light penetrates very slightly into the skin, not very far at all. Red light and infrared light in particular goes a long way into the body. And there's evidence, now this is quite interesting, that actually co-exposure to both sort of more blue and UV light and deep red light can actually offset each other. Don't quite know why. Now, I've asked some of my photophysics professors about this, and there is a theory that red light can actually do this something called reverse intersystem crossing. I won't go into it. I don't understand it, but the point about it is it suggests there are ways that this could happen. But another key point here is that we have to remember that everything is biphasic. I see in the literature at the moment, and certainly on all Amazon everywhere, there's a huge number of devices pushing out red light treatment. We know that actually too much is dangerous. And you talk to any sort of dermatologist about this, and there's a lot of evidence. For instance, certainly in the Middle Ages, with all the fires and the candle lights, people were being damaged by too much infrared because it was damaging internal systems, which I think is quite interesting. And the other thing to remember here, of course, is that each wavelength of light is different in terms of energy. UV and blue light has a lot of energy, and we know that it affects certain components of the electron transport zone in mitochondria, producing a lot of ROS. Green also affects one particular component of electron transport, and we know that red light, and this of course is the $1,000,000 question, is how is red and infrared light working? You'll probably see this thing called cytochrome C oxidase mentioned. It does absorb light in this frequency, but there's also something going on with water. Now, I haven't got time to go into this because it gets horribly complicated, but water order could be very key here. And this is science we don't understand yet, but it's turning out really interesting. So the implications on Earth, we evolved in the Goldilocks zone. We need to stay in that Goldilocks zone. At the moment, we tend to be overindulgent and we're changing all our buildings in the wrong direction. In terms of removing predation and infection, we've done quite well. We protect from the environmental. So the right amount of exercise, solution obvious, but not universally accepted. Life's generally lazy to see intelligence paradox, as I mentioned earlier. The right spectrum and timing light, not there due to living indoors and dry for energy, so in fact going the wrong way, but could be an economic case of cut cost of healthcare, for instance, in hospitals. Space implications, slightly different. We have to mimic the Earth's environment, and my suspicion is the problem is it's going to be financial. Artificial gravity is probably going to be one of the most important things we do, but it has a big cost implication because the cost of sort of lifting up a big station, for instance, all the tons and tons it requires and energy it requires. Although once it's running, it's probably quite cheap. But I've spoken to somebody within NASA about this, and their suspicion is that actually once you get one of these things up and running, it actually saves money because you then don't have to re-engineer everything in a space station, because without gravity, all sorts of things go wrong. You know, we obviously know the obvious ones, like going to the loo or where you breathe has effects, but they have to re-engineer these systems. Shielding from radiation. This is a difficult one. Yes, it's important, but as the recent Orion's capsule, which went around the Earth from the Artemis, showed quite clearly, yes, we could protect against astronauts within the magnetosphere, but when you went around the moon, it all fell to pieces because we couldn't protect as well as we thought we could against this GCR. And this is because this thing called secondary neutron generation. But so it's quite a big cost. Correct light spectrum. I think this again is actually an easily fixable one. Low initial mass cost, bit of energy cost, but it could be very good. Magnetic fields, technically maybe possible. How we can do it, not entirely sure. Correct the carbon dioxide. So it could be argued that technology and cost-wise, simply improving light conditions is the simplest. Maybe. Conclusions. In space, we need the artificial gravity, as we said, and the right atmosphere, etc. But there are two cost implications, the second of which may be easier, which light span is simpler. On Earth, increasing exercise and correct light spectrum are not that difficult. And it overall needs to be accepted that living outside of our evolved adaptive envelope will accelerate the aging process. And I think there's a thermodynamic explanation for this. On Earth, this is relatively easy to fix, where living a healthy lifestyle would likely offset the cost of improved lighting by reducing healthcare costs. In space, there is a clear cost health consideration, which to date has not really been addressed due to the fact that current astronauts are generally very, very healthy when they go up there. And this is something not everybody appreciates. In terms of photons, we actually do need to find a clear biomarker that measures how benefits come from the correct light. At the moment, we need to prove this with science. Final word, the photonic vitamin benefit. How much benefit would the correct photonic landscape be for the average person? An answer, it probably depends on where that person is in matching their evolutionary envelope to their environment and what we define as optimal health. Normal is not healthy. If the correct dose of each wavelength of light is a photonic vitamin and can offset to some degree a lack of environmental vitamins, e.g. exercise, that's exactly, we might be able to get some benefit. Health may be also organ specific. $1,000,000 question, how do we measure health? Surviving is not the same as living in optimal health, nor is it what we define as normal. And I'll finish on this. How do we define optimal health? Well, we've put this as can be defined for any organism as living in an environment that optimizes health span, ensuring morbidity compression at the end of life. With that, I'll finish. There's probably a lot of questions. There we go.
Amazing. Thank you very much, Alistair. So maybe I'm going to start off. I'm going to invite people to fire questions into the Q&A to get started. But you mentioned being outside the canalized envelope, and you're sort of talking about that in respect to space. But I would suggest that we're sort of living outside our canalized envelope in current times by spending so much time indoors and not, getting that right light spectrum. Can I ask you your thoughts on that aspect, maybe?
Oh, absolutely. I think you're completely right. Yeah, I mean, I think it's been, people don't even accept we have a flight envelope really, but it's I mean, Darwin would have understood this completely. But yes, I think you're absolutely right. We, in our pursuit of comfort, we are removing the very things that may be very essential. Now, some of them, like light, we can probably do without for a bit, but does it mean we're healthy? And the parallel story, as I said, is exercise here. Everybody think, oh, we don't really need to exercise, we're all healthy. Well, actually, no. And the answer is, once you start to think about it from that perspective, you start to realize that there are factors. And this is why I use the photonic vitamin idea. Yeah, you can survive for a bit without it, like you can do without vitamin C, although you develop scurvy. And as Geoffrey always likes the idea of, how you get scurvy on ships. Or Bob talks about infrared starvation, but it's under the same principle. So yes, the thing is, we just don't measure it at the moment. We don't
Mm.
know that living. I mean, if we were in a hospital, what is part of the problem with the hospital? Is it simply the light is not helping the patients? No one's even measured it. What would be the difference? So I think you're entirely right. We're just putting people in the wrong place to recover.
And so we've got first question in from Anna. Thank you very much for joining us, Anna, today. What kind of artificial light is currently used on board space? Well, let's say the space station, and in space travel. Do we know what the light
Yeah,
conditions are there?
yeah, they're moving because it's all about energy. Everything costs, and so what you try and do is what we're trying to do on Earth, which is to minimise the cost of light. So what they do is, and I think Bob can probably answer this better than I can, but I think they've certainly shifted towards LEDs because they're just more efficient,
Mm.
and it's probably as simple as that.
But then I'd argue that there's probably been a lot of research and a lot of time spent on developing the lighting for the space station. Maybe in a okay.
No, no,
Jeffrey's going no. Okay,
I don't think that's the case. I don't think people have
still,
thought about it like that.
okay.
But that's the point we're making. They haven't. This is the sad thing. If you look back through NASA history, they did a lot of investment in light research, and they even had a spin-off company developing infrared devices. And the originator of these devices said we need to take them on spacecraft. It was just buried, and they just stopped looking at it. So you tell me why they did that? I don't know.
The changeover, this was in 2012. The space station went from incandescent light to LEDs all in one go because it was thought that LEDs would use less energy, and none of these considerations were considered. And the same happened with submarines. So submariners now, when they're underwater for 67 to 70 days, or the most recent one was 200 days because the other submarine was broken down, they're in LED light for the whole period of time. So they're reds down, blues up.
Mm. So I was sort of kind of hoping you were going to say that actually the quality of light on the space station was at a higher level than what we have here back on planet Earth, but maybe not. And so again, putting this comparison between space and Earth, it strikes me that if we're spending 90% of our time indoors, or 93 if you're based in the US. Then really the only difference on that level is that we have a choice, and the choice is to step outside, and maybe stepping outside is slowing the the aging process ever so slightly. Whereas when you look at as an example, the two astronauts who came back after was it 6 months 9 months on the space station, their aging had been significantly accelerated, or yeah.
They looked at well, and this is entirely the point. Has anybody ever measured the aging process in astronauts? And the answer is no.
Yeah.
It's not even been a question, and this is the point we've come across and been coming across for 20-25 years with the sedentary lifestyle. Is people don't really see it as that. They see it as an increase in disease, not an acceleration of the aging. And I think that this is the point I would make from this talk, is that actually what we're seeing now in space is just an extension of what we see on Earth, is that there's certain aspects to health which are just not being looked at for whatever reason. And this is absolutely fundamental, because that's the zeit, as I said to you before, it's the zeitgeist of the time. Normal is normal. Oh, it's okay to be this, that, and the other. But when you actually look at it, well, maybe it isn't. And I think, and we certainly, I think Geoffrey and I have certainly come across this a little bit in the research we've been doing.
Well, we've suggested quite strongly to the space agencies with whom we've spoken that they need to assess astronauts' biological age before they leave and when they come back, as opposed to their chronological age. And it's now known that your organs age at different rates. And this is work from someone like Steve Horvath and others. And there are a number of ways in which we can assess that now. One is called the epigenetic clock. There's the proteomics clocks, where we can look at the actual age of, essentially your age of your mitochondria. And although the astronaut might look a bit tired when they get back, a few months later they seem to look better again, but their mitochondria may have aged 20 or 30 years. And that's the difference. In later in life, when they're going to need mitochondria to adapt to the onset of the normal age-related illnesses, they're going to suffer a lot worse than people that hadn't been in space. And we see the same, for example, with COVID. Our view is we haven't seen long COVID yet. You'll see that in about 2 decades' time, where we think COVID itself has probably had this similar impact on mitochondrial function.
Yeah, because we when the COVID hit, our first thought was, is there going to be a difference between fit and unfit people? And the answer was yes, and it could be to do with mitochondria, and that's why there was interest in people using red light. And we work with groups who are looking at red light and reducing inflammation in people with COVID. Yeah, and as Geoffrey says, I think we haven't seen the last of long COVID yet.
Yeah, maybe just sticking on COVID for a second. I seem to remember 5 years ago there was quite a lot of stuff about vitamin D levels, within the system, and how if you had a higher level of vitamin D, then you're more likely to be more resistant to the effects of COVID. But then as things have evolved, actually vitamin D was sort of being discussed as more of a biomarker for the amount of time you spent outdoors, and as a consequence were more resistant, and potentially that time outdoors was exposing you to more things like infrared.
Yeah, it's actually, the problem is it's a lot of confounders here. When you talk about the Swedish study, for instance, I mentioned, you can argue that, oh yes, well, people spent more time outside, got more sun, but there may have been the same people who just exercise more. So
the Swedish study actually balanced for those other factors. And the vitamin D, in a way, there is a thought that it is a biomarker, that actually the process by which the body is producing vitamin D, which is in response to photons arriving, is probably more beneficial for health than the end product of the vitamin D itself.
Yeah, but vitamin D, for instance, has been shown to upregulate mitochondrial function in muscle. And this again, people think of what does it do, but actually it's both modulated and controls mitochondrial function. The vitamin D receptor, for instance, modulates mitochondrial function, but there's also a direct effect, and indeed the mitochondria themselves degrade or modulate the vitamin D itself. So there's this whole pathway inside the body which nobody really looks at, which is, well, they do look at, but it's not as obvious as it is. And I think there's certainly something in the vitamin D story, because they were looking at vitamin D, and the problem was they looked at vitamin D, and there was a big thing, and as you said, during COVID, give more vitamin D and protect, and it didn't always work. But I think, as you suggest, I suspect simply just going out in the sunshine, and the point I was trying to alliterate a bit in the middle of it was, it's not just vitamin D. There's something else going on, and it could well be the infrared light itself is modifying the mitochondrial function in all your cells, including how it metabolizes and deals with vitamin D and melatonin. So there's more complexity here, but it's quite logical if you think about it.
But going out in the sunshine would be useful. I mean, modern building design should probably have a roof garden, where everybody is obliged to at least be outside 20 minutes before and after midday, and that would probably be all they need.
It seems that the intervention doesn't need to be particularly long. Alistair, you mentioned quite a lot about movement. You know, I sort of liken our artificial light environments today to nutrition, in the sense that, you know, if I have some fast food, for instance, I know that, you know, I'm going to be hungry about 30 minutes later. I've been fed, but I've not necessarily been nourished. You know, equally with light today, I think we're fed, but we're not nourished. um, and that's one of the challenges. You know, there's a series of elements. You know, and you mentioned movement, but yeah, I say I like the fact that we need light, we need nutrition, we need movement. Air and oxygen is clearly important, and you mentioned sleep and the fact that mitochondria might need to rest a little bit too. So there's lots of components that come together, that and as you mentioned, you know, the the continuous searching for convenience of lifestyle has weakened, you know, a lot of these elements here that we need. It's slowed down our drive for movement, our you know, access to light, all sorts of things.
Yeah, that's what I mean by the intelligence paradox, because it is predictable. It is predictable from the intelligence, because, you know,
Yeah.
you know, all organisms try and then there's quite a lot written in terms of evolutionary terms of this. All organisms try and modify their environment to remove the threats, which is quite natural. You know, trees do it. If we look at trees, they control the growth of other organisms around them to make themselves safer. You know, all organisms do it, and humans are really good at doing it. But the problem is that, I guess, in ancient times, it probably didn't matter so much, because you were probably going to die of infection or being eaten by something before you got to the end. But nowadays, it makes a huge difference, because it is, you know, it is having an impact.
Mm.
So
Sylvie's sent a question, and thank you, Sylvie, for the question. Will there be a change in the lighting industry to reflect the importance of infrared? Now, maybe that's more one for me to answer than for you guys. So I think that the challenge today within what we are doing is not a technological challenge. You know, the technology exists to do this stuff, you know, with relative ease. I think the biggest part of the challenge here is a legislative one, and we've gone down a very narrow focused pathway of lumens per watt, lumens per watt, lumens per watt, you know, ever trying to increase it. But as we increase lumens per watt, quality of light decreases. And yeah, there's no other way. But yeah, we probably need to spend a little bit more on energy. But actually, as Alistair, you were saying, you know, there's a massive cost burden over here, which is just getting bigger and bigger and bigger, and we're not necessarily taking preventative aspects into consideration at the front end. Yeah, I don't know if you've got thoughts on that balance between front end costs versus healthcare costs later on.
well, I guess, as we were saying before, this is the point we made years ago, when I was in Imperial. Actually, the point was made is that prevention actually saves a lot of money. But the problem is, I mean, there's a slide I've got somewhere which I showed, which when you actually look at the cost, and this was about 10 or 15 years ago, and when you look at the cost of you know, obesity and poor lifestyle, you know, it's up in the 80 to 90 billion pounds. The amount of money being spent by the government at the time on prevention was about 100 million.
But I think,
just doesn't.
Yeah, I think the issue is not finding new sources of red or infrared, is to just stop blocking the free infrared that's coming from the sun.
Yeah.
The important thing is to stop this sort of fascination we have with insulating, so you can't lose any heat whatsoever. But in the same process, is you're blocking an enormous amount of infrared. There's plenty of infrared. It comes from the sun. It's free. What we should do is allow it into our houses and into your buildings, as opposed to changing the lighting industry. The lighting industry, I think, needs to be mindful of this, and you need lighting that actually alters the red-blue balance throughout the day to give you a far better circadian. It's not a matter of switching on. I mean, one or two. Bob Fosbury will tell you that just put one red incandescent light in a room is probably all you need. That's a sort of starter help in terms of the fact there's no red at all. But once you are introducing red, the red-blue balance has got to be important as well for your circadian and the longer term health effects. So I think the lighting industry should deal with this. It means that it probably have to use more energy per lumen. But I think we've gone far too far. That pendulum has gone far too far now to the detriment of human health. But the first thing is make some windows that don't that allow the sun's infrared through.
Well, I think on that point, Bob did come up with a very, very simple concept, which is on all north-facing sides of the windows of a building, you use normal glass, and put trees on the other side, and let the tree, reflect the infrared back into the building. That way you manage the heat gain, the solar heat gain potentially, but you're still giving benefits to humans, as a consequence. Maybe that's a simple solution. There's a question from Lorraine. Thank you, Lorraine. Nice for you to join us today. If mitochondria need rest too, and it uses light to create energy, then is that why it's so important to have complete darkness when we sleep?
Well, is it ever completely dark? And the answer is it's never completely dark. But it's a good question. I mean, the fundamental question, well, back to another question here. The fundamental question, of course, is what's the red light actually doing? And there's two ways of looking at this. The one way is to say that it's purely that it stimulates an adaptive response. It does something, it creates a small amount of free radical, which stimulates the cell to adapt. Or it's actually altering metabolism, altering the way things work to make it more efficient. And is are cells dependent on red light reducing energy? This is a really deep question. I don't think it's one that's been answered yet. So the question would be, I think, does the infrared? I mean, how much infrared light is in a darkened room? That's a good question. The answer is I don't know. Because I mean, Bob could answer that if he's there. Because how much would you get at night? For instance, how much do you get coming through the light of the windows or at night into a darkened room?
We'll see if Bob drops in a response in a second. But
yes, it's a good question. Yeah.
Yes. Nicholas from UCL, who's researching sleep and near infrared, has an optimal spectral irradiance of near infrared effects on mitochondria been defined as yet. In regards to wavelengths, I've read that the peak sensitivity of cytochrome C is around 835 nanometers. Do you think it is more likely that a broader spectrum is more likely to impact ATP production rather than a specific wavelength, i.e. in the way that pineal melatonin responds to 480 nanometers?
Okay, there's two components. One component is, what is the right wavelength? And I think, having spoken to Bob about this a lot, I think it's much broader. There are clearly, water absorbs at certain frequencies, and particularly up above 1,000. But there are also other windows in there. For instance, how, to get into more technical terms, how you vibrate the hydrogen bonds, for instance, in a water molecule, could be important. We know that, as you're absolutely right, cytochrome C does absorb light. Now, whether that process stimulates its function or inhibits its function or does something else, we don't exactly know. There is a competing hypothesis by somebody called Andrew Sommer. It says it's nothing to do with cytochrome C oxidase. It's actually all down to water order. And something I didn't go into, and is that there's evidence, and this is kind of the link to the charge. And you remember I mentioned this idea of charge across a membrane. Now, the mitochondrial membrane has something like 30 million volts per meter across it. It's the same as the bolt of lightning. That does things to water. The water is polar, and especially when it's near a charged surface, you start getting water structure. Now, there's evidence that some of these structures could start to absorb absorb light in certain frequencies. So whether that is a process, there's also an idea knocking about that, for instance, infrared light stimulates or helps ATPase, the little rotatory motor, which makes the ATP from the proton gradient spin faster. It's like greasing the wheels. Is it doing that? And I think there's a lot of basic quantum mechanics here that we do not understand yet. And so it's a good question, but it's pretty open at the moment. And I don't think there is a single frequency. I suspect it's more than one. And if you look at the frequency of light that comes through, I think you'll find there's more going on with more frequencies. Because biology would have adapted to what's there.
It's possibly even more complex than that. That is, the frequencies will be different dependent on the stresses to which the mitochondria are trying to adapt at the time, possibly different for a different time of day and at different latitudes, depending where genetically you started your life, whether it was in the Rift Valley in Middle Africa or further north. These things are really quite complex. The other thing is that yes, the pineal gland does produce melatonin, and when I was at medical school, I was told that was the source of melatonin. It's now pretty well understood that most of your melatonin comes from mitochondria, and therefore the stimulation that would be outside the 480.
Yeah, that's the point I said. There's that paper where they suggest that NAR actually stimulates production of melatonin through the body. There is some scientific literature supporting this, and so how it's doing that is actually quite interesting. But quite often, one of the reasons you get production of melatonin, and this is perhaps a clue, is it's the same with UV, for instance, or any other light source. If you have enough of a stimulus, and this is back to this concept of hormesis, the chromophore, just say, for instance, there is a particular chromophore. If it's say it's green light and complex 3 in the mitochondria, it'll absorb light. That energy has to go somewhere, and that energy is a signal, and that signal will then tell the system to do something. And generally, it means it produces reactive oxygen, and which is actually the most fundamental signal in the cell. So in terms of melatonin production, the argument could be made that NIR is simply messing with a certain part of the structure of the mitochondria. It says, oh, look, we're being messed with, we need to change, we need to adapt. And the signal for that could be ROS. We know infrared light can generate ROS in mitochondria. So, and ROS is the most, this is, people think, when you hear about mitochondria, people say, oh, well, they produce free radicals and it's very damaging. No. It's complete wrong end of the stick. They're a part of the signaling process. And infrared is sending a signal saying, oh, look, something's happening here. We need to generate more melatonin to protect ourselves. So it could be something like that.
So I think, Geoffrey, you were talking about when you were studying, as a lighting person, I think I got quite indoctrinated into the concept that melatonin was, again, all produced by the pineal gland. And then it was in 2019, I think, when I came across Scott Zimmerman's paper, which is the melatonin, the optics of the human body, that suddenly you start to understand that yes, there is this other subcellular melatonin which is connected to the mitochondria, and all of a sudden the world starts to expand somewhat. So yeah, it is fascinating. I've got another question from Anna, actually. Do you think it's a mistake to legislate on light with bans on some technology based on lumens per watt and not taking wider health impacts into account? And I'm not going to take that as an industry one. I'm going to throw that one back to you guys.
Absolutely, she's absolutely right. It's a complete mistake. It's just if you think of some of the earlier industries where after a while we thought it was an excellent thing to do, but it damaged people, like the ladies that used to lick their brushes to paint luminous dials on clocks. That was thought to be an efficient thing to do, but of course they then all got tongue cancers, or the people that worked in paint factories that got lead poisoning. This incessant drive to achieve more lumens per watt is having unintended consequences. And I think that before anybody pushes any further to do that, they need to understand what the early, immediate, early and long-term impact is on human health.
Amazing. We've got an anonymous question. When mitochondrial dysfunction occurs, either here on Earth or in space, is it possible that the damage can be fully reversed? Or are the interventions such as lifestyle factors and building design coming back into the Goldilocks zone, more like bandages in preventing any more damage?
Good question. Right, there's two ways of looking at this. There's one that you induce permanent damage, and for instance, you damage the mitochondrial DNA, for instance, so you can't get the correct mitochondria, or you simply induce turnover. And one of the things the body is extremely good at, if you let it, is regeneration. And mitochondria, they're not just little static things. If you go onto Google and put in Amazon and mitochondria video, they're moving all the time. And what actually happens is they're constantly being regenerated. And not only are they being constantly regenerated, but their function is changing. and even within the cell, they have different functions. And this is actually a key point. And so when we talk about mitochondrial dysfunction, and this is something which comes into cancer, people say, oh, it's dysfunctional, it's all gone wrong. Actually, what you're seeing is an adaptive response. The mitochondria are trying to adapt to a local situation, so they're doing something differently. Now, that may not be the right thing to do, but it genuinely reflects something else going on in the cell, which needs to be corrected. And so in terms of the exercise thing, what we do know is that actually you can have this, what we call them dysfunctional mitochondria. There are cases where the process which removes the damaged mitochondria is inhibited. Now, in particular, in inflammation. And the reason for that, it's very ancient, is that the thinking here is that mitochondria, as they say, were once bacteria, and therefore there was this kind of immune challenge going on. And so the cell has evolved to deal with them through this idea. But we know in inflammation, for instance, that mitochondria quite deliberately, the duff ones, which are producing all the damaging reactive oxygen, are left because they generate an amplification inflammatory signal. What we know can clear that, and have you heard of senolytics? No, right, okay. There is a big area. Well, no, it's a big area of research at the moment in stimulating this process called autophagy. Now, this is how the cell cleans up its garbage, and there's a particular process called mitophagy, which, when activated, removes the damaged mitochondria, because they are producing ROS and they do damage, but the cell is very good at generating new ones. What seems to happen in some of these conditions, especially if the cell thinks the body thinks it's infected, you get inflammation, and this is like a vicious cycle. And you can end up, and this is what happens in the metabolic syndrome. You end up with a load of mitochondria which think they're in an inflamed environment, and so the body, instead of removing them, leaves them there to amplify an inflammatory signal, and that just makes it worse and worse. But what we do know, there are compounds, and there's some natural products. Quercetin does this, is it induces the cell to pick up its autophagy garbage removal system and then generate new ones, and this actually can completely reverse the situation. And so you can end up being very inflamed, but actually you can reverse it. And we see this in cancer too. So the answer is yes, you can regenerate if it's done the right way. The other part of the question was can you protect? And NASA are certainly looking at, for instance, in antioxidants and things, and particularly mitochondrial protectants. But I think that's a bandage.
I think what we have to think about, though, is that whilst mitochondria might be regenerating, getting better, there are all other systems that take advantage of the poor mitochondrial function. So it's like a bit of a slippery slope in a way. And we know, for example, in post-viral syndrome, mitochondria really aren't ever getting back to their norm. Certainly what people are calling about long COVID. And we're also seeing where mitochondria are part and parcel of the initial defense against or dealing with viruses, because we're seeing an upregulation of viruses that have been sitting in the body for a while. So we see this with Epstein-Barr virus, and that's seen not only post-COVID, but we're also seeing that in the astronauts. So whilst the mitochondria are on their knees, all sorts of other cascades are beginning to start.
They're not necessarily on their knees. It's like having your own army. You're doing is saying, right, actually we need the territorial army to come in and start shooting things. But the problem is you don't know how to switch them off.
Yeah.
And that's kind of what's happening. And I think so these processes are certainly in many cases are reversible. And we know that senolytics slow the aging process. And so basically it's all about vicious negative spirals. The body has these amplification loops. It can amplify an inflammatory signal, and if you can't switch it off, you end up in a really bad state. And that's basically what Geoffrey's saying. Viruses can do this. So
that's one of the things that we've considered for quite some time, and you'll see it in some of our lecture series, is the thought about mitotuning, is to retune mitochondria. And we're thinking that photons may be extremely important, the use of photons, of trying to pump certain aspects of the electron transport chain to get the mitochondria to retune them. And that may be the case, for example, Alistair showed a slide with some effect on breast cancer cell lines and the change in the onset of senescence. the thought might be for the future is to tune the mitochondria so that you don't have to kill cancer cells, but just push them back to being normal again.
Well, this is where this is an idea. I mentioned the morphogenetic field idea and this work by Mike Levine. And what we think is that actually one of the things that happens with cancer cells, they stop talking to their surroundings because they change their field. And we think the mitochondria, and this is a big section, that may well be important in this process. So if you retune them, the cell suddenly becomes cooperative again. And this may be a way that we can do it. And certainly with the light, you see what you could be doing, for instance, with the red light, you could be inducing an adaptive response to regenerate parts of the mitochondria, switching off the inflammatory bit and saying, oh, regenerate now, we don't need all this reactive oxygen anymore, so let's have a more functional mitochondria. The whole process is incredibly plastic. And I think that's the way of thinking about it.
So we've got a question from David, and actually David posted a little while ago. So a fascinating talk. Many thanks indeed. Lots to take in. In terms of spectral output of artificial light sources for interior environments, what would you, Alistair, recommend? And would it be as close to daylight as possible, or something different? Also, would it be possible to distribute the slide? Yeah, he's asking whether you can distribute the slides after the presentation. So whether you're happy to share the slides. But anyway, so first let's go back to the questions in terms of artificial light sources in the interior environments, in terms of spectral output, and whether it needs to be as close to daylight as possible, or something different.
Well, I'd say, I think that if you think about the evolution of circadian thing, then yes, your light in your house would mimic what's going on outside. So, I mean, I think this has been seen. I see many places talking about this. So you mimic the rising sun, then you have the midday within reason, and then in the evening you go to what is effectively down to starlight and moonlight, with and you mimic that thing. And certainly, I think but you have to think temporally here. You can't just have one frequency fits all, one spectrum fits all. You have to mimic what the sun is doing through the atmosphere as the Earth rotates. I think
it would be latitude and date specific as well.
Yeah, there's an interesting point here, is whether or not actually different people from different latitudes would actually be affected in different ways. I mean, if you've ever been down into the equator, the sun going down is incredible. It's splat. If you go up north, it never goes down. So that actually has an effect too. And I suspect the populations who spent thousands of years in these areas will adapt, will have different requirements, and especially the rate that it changes. Is quite dramatic.
And so, would you be happy to share your slides afterwards?
Let me have a look at them. I think most of them should be all right. I need to see. I haven't got any proprietary information in there.
And then Sana, hello Sana. Sana posed a question: do you anticipate the actual the lighting production industry will be the agents of change when it becomes apparent that there is a monetary value in adding in the different colors of spectrums in the right amounts?
Well, who else would drive it? The question I'll go back to you at.
So yeah, I'm gonna, whilst you mentioned, well, we spoke earlier anyway, and there is a prolific amount of product today. Let's stay within the sort of photobiomodulation or red light therapy arena. Now, whilst the science, isn't fully, let's say, understood as yet, I think there's a number of aspects here, and one aspect is that there is a better potential public understanding of light in a therapeutic sense as a consequence of this industry and coming about. What we haven't quite managed to translate yet is that those devices are, again, just a supplement, in the same way as we take vitamins as supplements because our nutrition isn't delivering enough. So I think there's a number of different aspects. I think legislation has a part to play. I think consumer demand has a part to play. I think the built environment, the designers and architects of the built environment and lighting designers have a part to play in this. I think it's not just one element. I think the let's say the lighting industry I believe should be taking a stance, absolutely, and should be leading the way to a certain degree. But there are other parts within the whole ecosystem that need to play a part of this, you know, including legislation.
Mm.
I think you're right. What would be good to see is a sort of moratorium at the moment on any further advances of squeezing any more lumens out of a watt until such time as there is some fundamental and basic information and understanding. There simply hasn't been enough research in this area to be able to set about mitigating procedures. I think that there is a danger that the technological advances will keep going ahead. We'll end up getting lots and lots of light out of minimal amounts of electricity, whilst continuing to damage human health. And I think what the lighting industry probably needs to do is to invest some time and effort and money into researching into the health aspects, and then start to very carefully alter the product offerings. But as I say, the best thing to do is just to ensure that whilst the lighting industry is trying to work out what to do, is get the glazing industry to stop filtering out all the infrared.
It does seem to come back to, you still need the data. I think you need the solid data.
Yeah.
You need solid, good, well-controlled studies which show quite unequivocally, using the right metric, that the suggestion you're making absolutely does work and does have a significant health benefit, measurable health benefit. A lot of it's hearsay at the moment. There's no, as I was saying as I said to you earlier, about the potential, could we have two wards in a hospital where you control, one is controlled, one is treated, you have the right light spectrum. I know you have to get it ethically done, but the point I'm making is you need something like that. You could do it. I mean, mice are difficult because, of course, they have different diurnal rhythms, but you need to do it on humans, really. I mean, it's possible to do it, I guess, but you'd have to have the right metric. So how would you measure it? Mitochondrial health? I don't know. It's just inflammation. Yeah, I mean, there are a number of ways you could probably do it, but you need the money to support the trial.
Alistair, what about just biologically aging people who are working in these modern light and glass environments compared to those that aren't?
Presumably that's taking measurement of mitochondrial health.
Well, there are a number of things. You could do epigenetics, but I think the problem with this is you have confounders, because, as I said, you can have people who you may spend more time in one place, but they might be doing something else, like eating more Weetabix or something, or running more, which completely confound the result. And you need to control for all that, else it's meaningless.
Or two identical buildings, one with one set of lighting and glass, and the others with a different.
Yes, absolutely. Yeah, well, something where you actually outset and you take blood from people and say we're part of a trial, and then look at it and see if there is any difference, because there are a lot of the markers you could get from blood which would show a difference. And if the populations are big enough and they're similar enough, then you can start to make significant. You can actually use statistics to show there is a difference. That would be quite convincing, but you probably need, I don't know how many people you need, Geoffrey, 100, 200 per building. They'd have to be an identical population and types and everything, so. This is the problem with science. You start getting into some complex experimentation.
You could do multi-building meta-analysis, perhaps, but I mean, they've got to start somewhere. It seems to me that not much of this work has been done anywhere.
Yeah, I mean, I think
we've just charged ahead, changing the working environment or the living environment, almost oblivious to the potential health impacts that might have.
So that leads me on to Scott Zimmerman, who has joined today. Thank you very much for joining, Scott. And as I mentioned before, Scott wrote worked with Russell Reiter on the study of melatonin in the optics of the human body. So Scott's just posted, great talk. Scott's based in New Jersey, so they're facing the Department of Environment petition, or who in the States are talking about increasing the lumens per watt to 125 lumens per watt for a retrofit lamp, which is going to be a disaster. So he's first of all saying, please support in Europe. But Scott's been looking at melatonin and cortisol biosensors, which are, he's been sampling, and he's seeing amazing sort of results from that. But he's also then commented on the glazing, which is presumably in the States is unfortunately controlled by local regulations, and equally over here is controlled to a certain degree through building regs and solar heat gain, as we've been talking. So, they are problems we're facing, everywhere around the world, it would seem. This is not unique to the UK by any stretch of the imagination. It's a global issue.
Yeah, I think that what you have to remember is we're horizon planners. We're looking over the horizon in terms of research. If you remember when Sir Richard Dole first announced that he thought that smoking caused cancer, it took 30 years for governments and established institutions to accept that and act upon it. So knowing there's a hazard and a problem, especially if it alters the regulations and then the long-term investment that industry has made, what we know now, or what we might find out in the next, say, 2 to 5 years as the impacts of light environment on health, that might take 20 years to percolate through to the sorts of light bulbs you put in your room and what type of glass you have. I mean, one has to accept that. But if you don't do the research first of all, then what we may have to accept is 30 years from now looking back saying, oops, we somewhat overlooked this.
And if he's just put in a post, which are some of the circulatory markers, GDF15, for example, as a question. I have no idea what GDF15 is. So Iffy, thank you very much for the question. I'm that's way out here for me.
To answer that question very simply, you could just look at inflammatory markers. I mean, there are loads of inflammatory markers you could use. These are very standardized tests you could look at. If you wanted to get more complex, you could certainly start isolating blood cells too. Even platelets can tell you information.
I'd look at platelets. Ready source, look at platelets, very active, and look at mitochondrial function. Then say hello, Iffy, and hi, Scott. Good to see you both.
So that's easily fixable. That's an easy one. Yeah, it's getting the experiments planned. But I think Geoffrey's right. You need the data, and with data you can do a lot, but it has to be solid. And we've seen this, for instance, with the light itself when we're looking for biophotons and things. And the big problem we found is that when you start to read a lot of the papers, they don't mean anything because they just weren't well controlled. The experiments weren't very good, and the general scientific community go, well, they're not very well done, so I don't believe it. And so the hurdles, and Geoffrey will know this better certainly with the cannabis story, the hurdles are very high, and your data has to be absolutely spot on. Then people will believe it. People are very skeptical, especially something like this.
Something you can't touch and feel.
I think what you're saying, what we're saying is, the key here is the data. But maybe one approach is to raise some funding to do the experiments. There must be enough people out there who are interested to do this, and then set somebody up to do this and to come up with some really good experiments. So you can go back to whoever ever the people are who make the decisions and say, well, look at this. This is real.
We'd be happy to help with that.
Okay, so there are no more questions. I'm going to start wrapping up a little bit. Geoffrey and Alistair, thank you very much for your time. It's been really wonderful. Actually, I'm going to say, Alistair, you've referenced biophotons a few times, and I know it's an area that Iffy has been working on quite a bit. So maybe that will be a talk for another talk later on down the line. Understanding that within our biology, there are photons being generated, which is somewhat mind blowing in a certain sense.
Right.
And actually, Sylvie, you part of your question that you asked about earlier, which I didn't actually fully answer, I don't think, which was will there be a change in the lighting industry to reflect the importance of infrared light? The International Association of Lighting Designers published a white paper just recently looking at a more complete spectrum of light for interior environments. And actually on the 5th of June, we'll be talking to Kevin Shaw, one of the authors of the paper, and hopefully a few others, as another Meaning of Light talk. That'll be the 10th Meaning of Light talk, but we'll start to examine where organizations like the International Association of Lighting Designers are sort of moving in their thoughts as well, which is nice to see that we're moving beyond just the visible. So, but back to you, Geoffrey and Alistair. Thank you so much for the talk, the presentation, your thoughts, sharing your thoughts. It's been really, really fascinating. Alistair, if you can have a look at your slides and see if there's anything that is you'd like to remove, and I think there are people who'd love to have a set of the slides too.
Okay, yeah, it would be a PDF. It wouldn't be the original PowerPoint.
That's absolutely fine. That's absolutely fine.
Thank you very much indeed, and thank you to your audience for listening to us.
Thanks for inviting us.
I think we've got a number of comments coming through now. Fascinating, extremely interesting, amazing discussion. Lots of people have really enjoyed it. So thank you very much for giving your time this afternoon. It's been an absolute pleasure listening to you and talking to you.
You're welcome. Okay, bye bye.
Thank you.
Bye-bye, everyone.
Take care. Bye bye.